Perfect.
Excellent. Sorry about that glitch. Let's go back to the data. So I'm taking here an evidence that physiological oxygen levels that we measure in vivo are critical for any experiments done with cells in vitro. This is taken from an elegant review by Jeremy Ward in BBA in 2008. And on the y-axis of this left-hand graph, what I'm illustrating from 0 to 25 is the partial pressure of oxygen in kilopascals. And at sea level, we can equate this almost to percent oxygen. So when we look at the air we breathe, we're breathing around 20.9 kilopascals of oxygen. And as we pass to the alveolus in the lung, it drops to around 13, the arteries, the small arterioles. And then when we come to the exchange bed of microvascular organs, such as the heart, which will be the focus of today, note that the actual oxygen level is way below 5 kilopascals of oxygen. Over here in this schematic, I'm just illustrating the point one more time. We have the air in the lungs that we breathe around 21 kilopascals. As we move into the lungs just here, it's around 20. In the alveoli, where we have oxygen exchange, it's around 13 to 14. And then when we go to arterial blood, I'd like you to really focus on this particular panel. Note that the oxygen levels depicted here range from 5 to 13 kilopascals, 13 kilopascals for the lung microvascular beds, and 5 for microvascular beds, for example, in the heart, the kidney, the brain, etc. Then when we move down into tissues and the cytosol of cells, this can drop independent on the organ of interest to around 1 or 5. And within the mitochondria, we know well that the oxygen levels can range between 0.5 and 1.3 kilopascals. So the real take-home message in this slide is, if you culture your cells in air in a standard 5% CO2 incubator, you are exposing them to hyperoxia. And that'll be the theme of what I'll share with you in the next 20 minutes or so. So let's focus on the heart. And this is taken from a review that Tom Keeley and I wrote for Physiological Reviews, defining physiological normoxia for improved translation of cell physiology to animal models and humans. But let's zoom right down on the coronary artery, where in humans, that's the red coating that you can see here, the blood has an oxygen level of around 5 kilopascals. That's this red artery here in the coronary artery. If we go to the myocardial tissue itself, in other words, this tissue here, and we look at the superficial layers, it's around 5, and the intracellular levels in an active myocardium, the blue, can be as low as 1. Now, how do we do experiments in cell culture? Well, let me just show you the schematics and the model, largely due to the generous support of Baker Ruskin and their Cytive duals. What you can see here is Tom Keeley and Salil Srivastava working in one of our dual Cytive workstations. These are oxygen regulated, 5% CO2 with humidity. They have a small Etaluma fluorescent microscope in here, and all our cells are cultured long-term in this defined oxygen environment. They never are taken out and exposed in a laminar flow hood. This enables cells to adapt to the oxygen level that we're interested in, and you can see here one of these flasks being viewed on the Etaluma microscope. The advantage of this Cytive workstation is that it has 2 chambers. And as you can see here on the left-hand side, we're maintaining that chamber at an oxygen level of 5 kilopascals. I'll come to that in a little bit later, but I already alluded to the fact that this is the oxygen levels that microvascular beds in organs see in vivo. To simulate ischemia reperfusion injury, for example, at a cellular level, we can have the opposite chamber at 1 kilopascal, which is hypoxic. So cells can be moved either from this chamber to that chamber, or what we can also do is use another device that I'll show you in due course from B&G LabTech to look at a oxygen regulated plate reader to simulate normoxia to hypoxia. The other thing that you can do so you don't use these workstations fully clogged up with cells, you can culture the cells in a Condacell device, which is oxygen regulated, maintained at the oxygen level from the chamber that you're working in, and then maintain them further in a tri-gas incubator at that correct oxygen level. Take them from the tri-gas incubator back into the respective chambers of the workstation. That way you avoid taking cells into oxygen environments, air, in other words. So let us look now, how would we predict in cell culture the oxygen level in a cell monolayer? If we were to set the headspace here at around 5 kilopascals oxygen to simulate what has been measured in vivo, and we looked at HeLa cells, umbilical vein endothelial cells, or contracting cardiomyocytes, you will note immediately that the umbilical vein and the HeLa cells are quite similar to the ambient level of oxygen in the workstation. But if you go to a contracting cardiomyocyte, consuming oxygen, you'll note immediately that the oxygen levels under this ambient headspace of oxygen in the workstation is markedly hypoxic. So you have to take into account the metabolism and the metabolic rate of your cells. Now, one of the important things we need to do is to use this device. It's an oxygen regulated plate reader. You can put a 96-volt plate that has been equilibrated or adapted in the Baker Ruskin workstation, and there's an oxygen control head that can control that. If we look at this graph here, we can use a nanoparticle phosphorescent probe, MitoExpress intra, to determine intracellular oxygen levels in live cells in culture. So we can incubate them in this plate reader at 20% and then drop the oxygen rapidly. And you can see as we drop the oxygen, the MitoExpress intra signal increases. And this is just to show you here a picture or an image that we take from the software of this machine, and we can measure both the ambient oxygen level in the plate reader as well as the intracellular oxygen level in cells. And that allows us to determine real-time oxygen levels in living cells under defined ambient headspace in the workstations. So what I'd like to show you here is we have our endothelial cells, in this case, umbilical vein endothelial cells, in the plate reader under room air, that's 21% oxygen. And you can see here the medium level is around 18% oxygen here in that humidified 5% CO2 incubator gassed with room air. And the circles show you the actual concentration in the intracellular space or cytosol. As we lower the oxygen to 5%, we get a very rapid decrease in both the medium, the open squares, and the circles, and you will see it achieve somewhere around 3.7% oxygen. Now, let us look here at a human umbilical vein in vivo. These have been measured in the blood to have an oxygen concentration of around 3.7% or 3.7 kilopascals with flowing blood. And this is how most experiments are conducted in room air with cultured cells. And you see that the intracellular oxygen concentration approximates around 16 to 17% oxygen. Remember this message. Never does a cell in vivo see 16 to 18% oxygen. Absolutely never. When we come and try to recapitulate this level measured in vivo, if we gas the workstation with 3.7% oxygen, note immediately that we have a lower oxygen concentration than that actually in the workstation or the level measured in vivo, 3.7. So we have hypoxia. When, however, we take account in cell culture monolayers that there's a gradient of oxygen so that the endothelial monolayer does not actually see 3.7%, fortuitously, we gas the workstation at 5%, and we're able, by serendipity, to achieve the exact intracellular oxygen concentration that has been reported in vivo. So it's really important that you understand the possibility of an unstirred layer or an oxygen gradient in cell monolayers that don't see flow. Now, one of the critical things that I'd like to point out here, and I'd really like to draw your attention, please, to the left-hand slide first that I'm highlighting. When you lower oxygen in cell culture, you will activate or stabilize HIF-1 alpha. And here is data that we've had cells adapted for 5 days to 18, 10, 5, 3, or 1% oxygen. And what you can see is that very negligible effects on HIF-1 alpha expression versus beta-actin. And only when we are at 3 to 1% oxygen do we begin to see the stabilization of HIF-1 alpha. If we plot the data against the intracellular oxygen concentration, that's the gray curve, you'll see that as we lower oxygen, we're only beginning to get stabilization of HIF-1 alpha well below 2% oxygen. In other words, we're beginning to see the physiology in cell culture. On the right-hand side, I'm showing you a schematic, which I'd really think is the take-home message of this slide. We're plotting on the y-axis protein expression of HIF or messenger RNA. And what you can see as we lower oxygen from 18 down to around 3.7% or 5% oxygen, we would get an initial rise in messenger RNA for HIF-1 alpha, which stays elevated over 4 to 6 days. And the protein expression will peak somewhere around 2 days and stay elevated for up to 4 to 5 days. That is why we incubate or adapt our cells for at least 4 to 6 days in a defined oxygen level to minimize the activation of HIF-1 alpha signaling pathways, since we're interested in physiological normoxia. Now, is all of this important? I believe it is absolutely critical. On this axis here, taken from a paper by Tom Keeley et al. In 2018, we're looking at cell viability. This is looking at active mitochondrial dehydrogenase activity on the y-axis, and we're increasing the dose of ionomycin. This is a calcium ionophore to simulate calcium overload in these endothelial cells. Note the open circles are cells adapted to 18% oxygen. You can see the marked decline in cell viability. When you pre-adapt cells for 5 days to 5% oxygen, the level in vivo, note the protection that you can see at a given concentration of ionomycin. If that's not convincing, we've used an Nexin-5 staining, that's for the phosphatidylserine groups damage, and you can see that in cells at 18%, ionomycin leads to a huge damage. That's the red in Nexin-5, the blue are DAPI nuclei, and at 5%, markedly reduced. And this is just the summary data to show the massive damage or an Nexin-5 staining, which is absolutely protected when you have cells adapted to 5%. So this is an important finding. And if you want more information on that, please look at that manuscript. Another really critical finding is the following. These are lung epithelial cells. I'm showing you here on the right-hand side, cultured in a standard incubator. In other words, around 21% oxygen. In this paper, what we were interested in is, do increasing doses of nanoparticles to deliver drugs to cells cause damage to the cells? Note here, we're looking at an intracellular antioxidants in nanomoles per milligram protein. As we increase the dose of nanoparticles, these cells in air are resistant to damage until you reach a very high concentration of the nanoparticle, where you now have cell damage and loss of this marker of viability. That's how most people do cell culture. When we take these same cells and adapt them to the level of oxygen that I alluded to in my beginning slides in the lung, note that the intracellular antioxidant level of glutathione, instead of being around 45, drops to 30. And secondly, now the nanoparticles are actually causing loss of cell viability or loss of intracellular glutathione. This suggests that these cells, because the glutathione level is higher here than here, basally, are under hyperoxic stress, and that the readout of a drug screening regime in industry here would say, go to the clinic, these nanoparticles are safe. In this one, under physiological normoxia, we note that these nanoparticles are not safe, as they're actually leading to cell death of these epithelial cells. So that's another take-home message. The other interest that my lab has is in redox signaling. It's a complicated slide, but I'll walk you through it. this redox sensitive transcription factor is normally tethered to its cytosolic partner KEAP1, ubiquitinated and degraded via the 26S proteasome. When cells in vivo or culture are exposed to electrophilic stress, oxidative stress, or nitrosative stress, we change the conformation of this partnership. We stabilize NRF2, and endogenously synthesized NRF2 now can accumulate in the nucleus, bind to an antioxidant response element together with a small MAF protein, and lead to the upregulation of antioxidant genes and proteins inside cells. I don't want to talk about any of this below. Just recall NRF2 translocates to the nucleus, binds to an ARE, antioxidant response element, to upregulate target genes. Now, we asked the question, what would happen in cells if we adapt them long-term to the right oxygen level? And this is what I'd like to share with you. Without going into too much detail, heme oxygenase is a target of NRF2. And here we're plotting the heme oxygenase protein level versus reference protein alpha tubulin. In air, standard culture, we get a robust increase in response to a lipid peroxidation product, in response to an electrophile, sulforaphane, and in response to an NO donor. As we adapt cells for 1 day or 5 days to physiological normoxia, in each case, the induction of this stress protein is markedly reduced, independent of the stimulus that you are activating the cells with. This tells you that under these conditions of hyperoxia, you get robust increases in antioxidant defense proteins, which are markedly attenuated for heme oxygenase under physiological normoxia. And you can read further about this in this paper. Why is this? And the answer is quite simple. If we adapt cells to 5% oxygen, and we now transfect them with scramble or a silencing BAH1, a negative regulator of this heme oxygenase expression, here you can see that if we stimulate with an electrophile, we get an increase in BAH1 protein levels. Sorry, we don't get an increase. They're not changed. But when we silence BAH1, they're markedly reduced. Under 5% oxygen, I showed you in the previous slide that the induction of HO1 by an electrophile is minimal, but with BAH1 silence, we rescue this and we can now see this. In other words, BAH1 is the negative regulator of heme oxygenase, and this protein is upregulated in cells in 5% oxygen. We see the same kind of phenomena in brain microvascular endothelial cell. HIF1 alpha is only stabilized in these brain microvascular cells under long-term hypoxic culture. We know that certain enzymes, such as catalase scavenging hydrogen peroxide, are reduced under 5% oxygen. Cellular glutathione, like in the lung epithelium, are reduced. And even the proliferation of cells is markedly different under 5% oxygen, the lower curve, versus the cell. What I try to show you in today's lecture really is the importance that most, if not all, of previous cell culture, including all of our published work, has been conducted under hyperoxia. Under these conditions, any stress agent will still lead to the induction of stress genes, very important stress genes here. But under these conditions, the negative regulator of these NRF2 target genes is not in high expression. So hyperoxia has lower levels of this negative regulator. But if we adapt cells to physiological normoxia, in this case, 5% oxygen, we still get nuclear translocation of NRF2 in response to stimuli. However, NRF2 now drives glutathione-related genes. I didn't have time to show you that data. And we believe that these genes are operating under physiological normoxia in vivo. But now the elevated levels of this negative regulator are inhibiting the induction of these genes, heme oxygenase, that I showed you during the course of the lecture. So in summary, and finally, please, for those of you that work in cell culture, come to us, seek our advice about how to run experiments in cell culture to recapitulate oxygen levels that are measured and encountered in vivo. And I certainly refer you to our physiological review article in 2019 for significantly more detail. And now it remains really to say thank you to everyone who works in the physiological oxygen laboratory. This is Sarah Chappell, the senior author of our free radical paper. This is Richard Xiao, whom you know. This is Tom Keeley, who's now working with Peter Ratcliffe. And here's Tom with Salil Srivastava. Over here is Ron Jacob, who contributed to calcium experiments in physiological oxygen. Emily Borman works with neuronal cells. Julia Loy with tumor cells. FANS data I showed you in the coronary artery smooth muscle cells. Alex Morel, Wolfgang Merritt, and Theodore Stewart, and also Charlie Beale, whose photo I couldn't find, were involved in doing the laser ablation ICP-MS data that I showed you. This is Matthew in one of our workstations. And this is Gabriella, who did the brain endothelial cells. And this is Paul Fraser, a longtime colleague with whom we published this paper in Redox Biology. And that shows you that we don't just work, we actually also try to have some fun at a meeting in Lisboa in 2018 before COVID. And for those of you, here's a YouTube, I'll leave the slide on. You can click this and it'll show you how to use one of these workstations. And with that, I'd like to thank you for your attention, and happy to take any questions in the questions and answer session that I can answer subsequently. Thank you very much.